In the world of genetics, life is a tapestry woven through generations.
DNA contains the instructions needed for an organism to develop, survive, and reproduce. To carry out these functions, DNA sequences must be converted into messages that can be used to produce proteins, which are the complex molecules that do most of the work in our bodies. The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicus unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase called a primase constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides. Cells in a multicellular organism also reproduce by cell division, but the new daughter cells that are produced are not two separate organisms. Instead, these new cells are just parts of the organism, allowing it to grow, or sometimes replacing cells that are worn. In a modified example of cell division, sex cells called gametes are made. Chromosomes are an important part of cell division. Sometimes, mutations occur, leading to diversity in life. These genetic changes can result from various factors, such as environmental influences, errors during DNA replication, or even the deliberate alteration of genes in a laboratory setting. Mutations are the driving force behind evolution, as they introduce new genetic variations into populations. While most mutations have no significant impact or are even harmful, some can be advantageous and provide a selective advantage in specific environmental conditions. Over time, these advantageous mutations can become prevalent in a population, contributing to the adaptation and diversity of life on Earth. Humans have harnessed genetics to create new possibilities. Through the field of genetic engineering, scientists and researchers have unlocked the potential to manipulate and modify the DNA of organisms, including plants, animals, and even humans. This breakthrough in genetic technology has opened the door to a wide array of applications and advancements, from genetically modified crops that are more resilient to pests and environmental conditions, to gene therapies that hold promise for treating genetic disorders.
Join us on this journey through the continuity of life in the world of genetics, where every gene tells a story.